Hello First Form. In this little short teaching video, we're just going to do a quick recap on our work on microscopes that we did before Christmas. I'm going to recap the different parts of the microscope, which you can find on page 2 of your notes, and it's really, really important that you learn the different parts of the microscope and what they are used for. And then I'm going to do another quick recap on how we view an image using a microscope and how we prepare a slide, all of which we did before Christmas, but we're going to do them with a couple of different examples and just to remind you of those processes. So First Form, in this video what we want to do is to recap on our microscope, the different parts, and how we use it to view an image of something maybe very small that we want to see in greater detail. So we've got our microscope here, and I want you to spend a little bit of time learning the different parts of the microscope. That's really important. So we'll recap those very, very quickly. Firstly, if you remember, we talked about how to hold the microscope. We hold it by the arm, or sometimes called the handle. That's this part here. It's important not to hold it by the lenses, particularly up here. It can be tempting to hold it by that, but that could break the microscope or cause a lot of damage to that. On the side here, our microscope looks a little bit different than the one in your notes. But you can see that we've got our focusing wheels or our focusing knobs. The bigger one is what we call the coarse focusing knob, and you can see actually I've got it on to an image here connected to my iPad, and you can see that coming in and out of focus, and hopefully you should see it moving the stage up and down as well. So that's the coarse focusing knob, and then the fine focusing knob brings it into greater detail. We'll look at that in just a wee second as well. Okay, then we've got our different lenses. So up here we have got our eyepiece lens. Now this is called eyepiece because it's where we would put our eye. Now we've got our camera here instead today, but normally we would put our eye, we would look into the microscope at this point, and this is called our eyepiece lens. Mostly they multiply the image by up to 10. So you can see here on the side it says times by 10, so that's the magnification of this eyepiece lens. So it makes the image 10 times bigger, just this lens. But then if you remember, we have 3 other lenses down here, and these are called our objective lenses, and we can use what we call the turret to turn these. Now I've got to make sure it doesn't make contact with my slide. But that will then bring it into a greater magnification. You can see the one over the image at the minute is the smallest. If I bring this right down, I'll be able to turn them all fully. This is the smallest objective lens, and it is, let me have a quick look, it's times by 4. So if the eyepiece lens multiplies it by 10 and the objective lens multiplies it by 4, that's a total magnification of times by 40. Remember, we multiply the 2 magnifications together to get the total magnification. So times by 10 times by 4 gives us times by 40. If I turn the turret, so that's this little bit at the top, that's what I use to turn the objective lenses, I can put it onto the next magnification. So the medium power lens is times by 10, so it makes the image 10 times bigger. So that would be times by 10 times by 10 is multiplied by 100 in total. That will be the total magnification. And then finally, if I use the turret again to turn it to the high power lens, that is times by 40. So that's times by 40, eyepiece lens is times by 10. That gives us a total magnification of times by 400. Okay, so when we're viewing an image, we always start with the low power lens first of all. Okay. This is the stage, okay, so it's where I put my slide, it's where I put the sample that I want to view. And you can see here I've got a little slide on my stage ready to go. And these are simply called the clips, okay. These are the clips, and they hold in the slide in place. Okay, underneath the stage, it might be hard for you to see in the video, but you have what we call the diaphragm, and remember we spent a bit of time in class learning the spelling of that. It's a little bit complicated. And then down below we have our light source, or sometimes, as we said, that can be a mirror reflecting light off a lamp, but that would have to be separate. And at the minute my light is off, so I'll turn it on and you can see if you can see a difference. on the iPad. So did you see that? I'll turn the light off again and I'll turn it on again. So you can see that brightening up. And it's really important to see an image at all. We need some sort of light source, either a light coming directly below the stage, as it is in this one, or a mirror that's reflecting light off another light source. So those are your main parts of the microscope, and it's really important that you learn them really well. So as I said, when we're viewing an image, we always start with the low power lens first of all, and we were able to see something a minute ago, but we can't quite see it now. So what we normally do is bring it up to as high as we can, because it's really important when we're focusing this that we don't allow the slide to touch the objective lens, because that can cause it to crack. So it's better that we make it as close as we can without them touching, and then focus away. So let's see if we can get an image. We can nearly see something there. And this is back to what we did just before Christmas. I'm just turning the focusing knob, the coarse one, until I can see an image. So if you remember just before Christmas, we had a look at how we could take a tiny, tiny letter from a newspaper clipping and see if we could work out what letter it was from the microscope. So hopefully you can see the main shape of it here. This is the letter H from a tiny letter from a newspaper clipping. And what I can do on either side is I can use the fine focusing knob. It might be hard for you to see at this stage, but that will bring it into finer focus. You can see it's getting a wee bit more blurry now. And there it's getting a bit more focused. I'm turning it the other way. So the coarse focusing knob we normally use to get the image into view in the first place, and the fine focusing knob then to use it to get it into finer detail. Okay, so hopefully you can see that that's the letter H. Now, let's see if I can put it on to the next lens. Okay, we've got to make sure when we're turning that round that again it's not going to make contact. If it does, then again we would make sure that that is the distance is increased so that they don't make contact. So let's see. Okay, that's as close as they can now without touching, and let's see if I can get an image into view. Okay, so can you see it there? So you can see it zoomed in even more. Will not fit as much in. Let me see if I can try and get a wee bit more of it in. So you can see as I move around there, there's the different parts of the letter. Okay, so there's the start of the tall bit of your H. But you can see that zoomed in, and it's a much bigger image now, and that's because I'm on the medium power lens, so it's magnifying the image even more. The first one we looked at was making it 40 times bigger. This one is making it 100 times bigger. So what I want you to do is to have a wee look at page 3 of your notes, and I'm going to actually switch this slide here. We're going to have a look at another couple of slides. The first one we're going to have a look at here is a slide of a moss leaf, so a leaf of moss. And I'm going to use it to help you see what happens when we move the slide about. Okay, so before I put that directly on, I'm going to wheel this right back down again, and we're going to start at the low power lens, because remember, as I said, when you're starting to view an image, you always start with the low power lens. So let me put that over so I to the point where I think we'll see an image. And again, what I'll do is I'll wheel that up so it's the smallest distance between the objective lens and the stage, but making sure they don't touch. And then I'll focus away. So I'm turning the coarse focusing knob until I see an image. Okay, can you see it now? So hopefully you can see our leaf. Okay, and I'll use that fine focusing wheel to get it into better focus. Okay, so hopefully you can start to see the little cells in the moss leaf. But what I want to do is to have a look at your notes and see this idea of what happens when I move this slide around. Okay, so make sure you're at page 3 of your booklet. And it says there in question 1, it's asking, using the low power objective lens to move the slide in the directions given, write down your observations. So I want you to have a look at the iPad and see what happens when we move it in the different directions, okay? So first of all, I'm going to move the slide to the right. Have a look at the iPad screen at what happens. Okay, which direction is it moving? I'm moving it gradually to the right. What direction did it move? hopefully you saw that that moved to the left. Could you write that into your booklet? That when we move the slide to the right, the image moves to the left. Let's try it the opposite way. The next one in your notes is what happens when you move the slide to the left. So if I move it now to the left, I'll try to do that as slowly as I can so you can see. I'm moving it to the left. What's happening to the image? Which direction is it moving? It's moving to the right. Okay, so when we move the slide to the left, the image moves to the right. Okay, let's have a look now at what happens when we move it up, or we'll get the leaf back in image. When we move it up, can you see that it moves down? And when I'm moving the slide down, what's happening? The image is moving up. Okay, so let me do that very quickly again. Let me get it kind of in the center again. Where's it gone? Okay, there it is. Okay, so from the top of your notes then, the first one says what happens when it moves to the right. So look very carefully. When we move the slide to the right, the image moves to the left. What happens when we move the slide to the left? The image is moving to the right. Okay, let me put it back in the center again. What happens when we move the slide up? The image moves down. And what happens when we move the slide down? The image moves up. Okay, so in other words, when we move the slide, the image that we see, it moves in the opposite direction. So let's just check that you were able to get everything okay from that video and fill in your notes on page 3. And what we were doing there was we were viewing an image on the low power lens, and we were having a look at what happened when we moved the slide to the left, to the right, up and down, and so on. So what happened when we moved the slide to the right? The object or the image moves to the left. Then when we moved the slide to the left, the object moved to the right. So that's what those little dashes means. Maybe you could write them in and fill in your own notes. So object moves to the right, but that's what those little dashes mean, just the same word as above. We maybe talked in the video about the next bit about moving it up and down. So if you remember, when we moved the slide up, the image moved down. When we moved the slide down, the image moved up. But that's a bit like saying towards you and away from you. So if you move the slide toward you, the object or the image would move away from you. And if you move the slide away from you, the object moves towards you. The next one we didn't talk about in the video, but you can have a think based on what we've seen so far. So what we saw so far is that when we move the slide, the object moves in the opposite direction. So if you move the slide in a clockwise rotation, if you moved it clockwise, what do you think would happen to the object? So if you move the slide in a clockwise direction, what would happen to the object? Well, yes, just as before, we see that it moves in the opposite direction. So if we move the slide clockwise, the object moves anti-clockwise. And likewise, if we move the slide in an anti-clockwise direction, what do you think happens to the object or the image that we see? The object, of course, moves clockwise in the opposite direction. So when you're moving about a slide, and it takes that little bit of working with it, a little bit of fiddling around with it to get that idea of, okay, well, how do I get my image in view? And it's playing around with it and manipulating it that you see, okay, actually, if I move it to the left, the image is going to appear moving towards the right. And hopefully someday soon that you'll get a wee bit of practice in using microscopes and seeing that for yourself. Then it said view the image with a medium power lens. So if you remember taking, for example, our letter from the newspaper, we then viewed it using the medium power. What did we see then? Well, we saw the image more zoomed in. The image, the letter appeared bigger, it appeared more magnified. So write something along those lines in your notes. So when we move from the low power lens to the medium power lens and then to the highest power lens, the image will appear bigger each time. The bigger the lens that we use, the more powerful the lens that we use, and the image will become more magnified. And we can even see that by the magnification moving from, say, times 40 to times 100 to times by 400 in total with the highest power lens. So I'm about to show you another video of me in school demonstrating another part of a microscope practical. We're going to look at how you prepare a slide, in particular if you want to look at a particular type of cell, a particular substance, like our newspaper slide. We looked at that a wee bit. But in this case, we're going to look at the cells in an onion, and we're also going to compare those to the cells in a moss leaf, which you've just seen in the previous video. So when we come on to this demonstration of preparing an onion, slide of onion cells, we're going to need to use a stain. So just to complete your notes here, but you'll hear it in the video anyway. The name of the stain is called iodine. And if you think about it, onion, if you think about cutting up an onion, it's white. But what we're going to do is we're going to peel a really, really thin layer that's actually transparent. And so therefore, if we viewed it under a microscope, we actually wouldn't be able to see any of the parts of the cell. And so what we do is we add a couple of drops of a kind of orangey liquid that will stain the cells and allow us to be able to see the different structures within the cells. So rather than us using water like we did for a newspaper to get the little cover slip to stick down and to coat the sample on the slide, this time we're going to use a stain called iodine. So listen up for that name in the video. So as we go through the next video, I want you to have your booklet open at the page after page 3. It doesn't have a page number on it, but just the next page. We'll turn to that now. So this page here, have this open as we look through the next video, but I'm going to demonstrate this. So first form, I'm now going to show you how to prepare an onion slide, and it's really important that you know how to do this. Hopefully at some stage you'll get to do this, but for now I'm going to demonstrate it. So what we basically want to do is see the cells in this onion. Okay, and what we need to do is to get a sample of those cells first of all. So I'm just going to cut off the top of my onion. And first thing I'm doing is really just getting a section of my onion, because if I was to put the whole onion in, it would be far too thick. I basically just need one layer of my onion cells. So I'm going to get right down to just one little segment of my onion, and then what I'm going to try and do is just peel off a very, very thin layer. This is sometimes easier said than done. But there you go, hopefully you can see it's starting to peel away. So just need a little bit. I've ripped a wee bit off there, so I'll pull a wee bit more. And then I'll maybe just cut that to try and get that last wee bit off. Okay, so let's see if I can get that without ripping it. Okay, so I'm not sure whether you can see that in the video, hopefully you can. But there is just one really thin layer, so not even just like a slice of onion, you're literally thinking one thin layer of the onion skin. Okay, so what I'm going to do is I'm going to put it on one of my slides so that I can see it, trying to get it as flat as possible. Remember we talked about before Christmas, sometimes we can see wee air bubbles in our images, and those are not ideal, and sometimes they block what we're trying to see. Okay, so you can see there, I'm just using my tweezers if it'll stick down. Okay, remember we want to try and avoid using our fingers, otherwise we can get fingerprints on our image as well, but this does not want to stick. The other side. There we go. Okay. So I want to just put that down as flat as I can, but again, trying not to get any air bubbles. Okay, now I don't want to just put that straight on my slide. Two reasons. One, I actually won't see anything because our onion skin and our cells will be transparent. So what we need first of all is a stain in this case, because our onion cells are transparent and they don't have a colour, so I actually wouldn't see anything if I put this under the microscope now. So what we use is a solution called iodine. Hopefully you can see that in the video, and you need to know the name of that, and that will be filled in at the bottom of page 3. The stain that we use is called iodine solution. You can see it's an orangey colour, and all it does is simply stain the cells so that they're easier to see and the different parts are easier to see. So I'm just going to put 1 or 2 drops on, and then I don't want to just put this under my microscope now, because it wouldn't be protected from the lens wouldn't be protected from it, I should say. If I was to put this straight under, the lens might get iodine on it, which could stain then the lens. So what I want to put on it is this little cover slip. It might be hard for you to see in the video, but it's just a very, very thin piece of glass. What I do is I hold it at a bit of an angle and then lower it down, and I can just use this, use my tweezers to do this. Sometimes use something called a mounting needle, and we want to lower it down very gently, okay, to try and minimize the risk of air bubbles. Okay. Now, hopefully you can see that is now done. I'm just going to wipe up a little bit of the iodine from the edges, okay, in case any of it spills. So now let's have a look to see if we can see these on our microscope. I'll set this up here. I'll clear this out of the way so that then we can get the iPad. And maybe you're glad that you're not actually here because of the smell of onion. We were talking about this, and having your bubble room smell like onion all day might not have been so nice. So I'm going to turn on my microscope again. I just turned the light off there. Hopefully you can see that it's at a good angle for you to see. And again, I'm starting with my low power lens. So remember, it's really important that when you're viewing an image for the first time, that you view it on the low power lens. So I'm clipping my slide in place using the clips. I'm clipping it on the stage over where the light is coming up. Okay. So I've got to use my coarse focusing knob to get my image in. I'm going to minimise the distance just in case that was to make any contact, and then I'm going to focus away. So hopefully now you can see our onion cells come into image. So that is what the cell of an onion looks like, and in a later lesson we'll spend a wee bit of time looking at the different parts of the cell, but hopefully you can see these outlines. This is what we call our cell wall. Now I want you to try and ignore these black circles because they are air bubbles, or the wee bubbles of our liquid, but I want you to see, can you see that slightly darker spot there? Okay, that's what we call the nucleus of our cell, and we can see that really clearly. Hopefully you can see it as well. So you can see that in most of the cells. Every cell will have a nucleus. It's like the control centre of the cell. Hopefully you can see that, and we'll look at those different parts in a later lesson. I'm going to now turn it so that it's a closer image, and we'll see if we can see anything in greater detail. So I'm just using the fine focusing wheel because you can see, I can see an image, it's just not very clear at the moment. Okay, and hopefully that's a bit clearer for you now. So again, hopefully you can see the cell wall and our nucleus. A plant cell, an onion is a plant, it will also have what we call a vacuole where it stores its food. Most plant cells also have something called chloroplasts, but onion cells do not have them because they're not green, and they are not used for that process called photosynthesis that we looked at a little bit. We'll talk a wee bit about that more at a later stage as well, but that's something unique about an onion cell. So what we're going to do now is we're going to draw what we see in this image in our notes. But that process of preparing an onion slide is on the page after page 3. And then at the top of page 4, we're now going to draw what we saw on the microscope. So what we're going to do on page 4 is to draw one of those onion skin cells, just one of them. You can flick back to the video that you just watched to see what they look like, and can you make it look like what we saw in the video. But we're just going to draw one. Now, as with other diagrams in science, I want you to do this in pencil. So if you could get a pencil out now to draw this, if you don't have one to hand, pause the video quickly, go get a pencil and then join us again. So you could see they were quite long and thin, and hopefully you saw this kind of shape, and they were all sort of stacked on top of each other. So something like that, some sort of regular-ish shape, but you could see that some of the edges were a wee bit diagonal as they stacked with the other ones. And then you could see that the wall, it was thick. It was, well, you could see it two-dimensional as such. It wasn't just one thin line. We could see that it was a thicker line. And that's because plant cells have what we call a cell wall. So I'd like you, again with your pencil, to draw a line out from that and label that as your cell wall. We normally label the outside line as the cell wall, because actually running along the inside of the cell wall is something that animal cells do have, and plant cells also have. We will have a look at this in a later lesson, but that's what we call the cell membrane. The cell membrane, and it controls what goes in and out of the cell. Now, plant cells will also have what we call a vacuole, but it was difficult for us to see that, so we'll perhaps leave that out for now. But what was really clear in our image of our onion skin cells was that nucleus, that darker circle. It was stained orange from our iodine, but we'll just draw it as a dark circle. And that's what we call our nucleus. Be really careful about your spelling for this. We'll focus again on our spelling of these parts in a later lesson. But it's spelled N-U-C-L-E-U-S, nucleus. So try and get it right now and get in the habit of spelling these things correctly. Again, don't worry too much about these terms for now. We're just drawing what we saw, and in a science diagram we always label the different parts of it. But don't worry about the different parts, what they do, what they are yet. We'll spend more time on that in a later lesson. And then the last thing I want you to label, which again is hard to see as such, but there's a kind of watery, thick watery solution that fills cells, and that is called cytoplasm. C-Y-T-O-P-L-A-S-M. Again, don't worry too much about that for now, but that's like the solution that fills the cells, and it's where all the chemical reactions of a cell take place. So from just cutting up an onion, taking a small section of the skin, placing it on a slide, and putting a little bit of orange stain on it, that iodine stain, covering it with a cover slip and viewing it under a microscope, we could actually see the cells that make up an onion skin, which is pretty exciting. And from it we were able to identify the cell wall, the cell membrane, the cytoplasm and the nucleus, so you can tick those parts that you were able to see. Just one last question to look at really quickly, and it's to think back from our onion cell and compare it to the moss leaf cell. Now, I'm going to show you some pictures here because I think it's probably slightly easier to see the differences. And we're just going to have a look at what's the difference between, the very visible difference between the moss leaf cells and onion cells. So here are some pictures, first of all, of our onion cells again, and here you can see the magnification is in the corner, and these are onion skin cells again in iodine. You can see that's very similar to the image that we saw from the microscope that I did in the lab. If we get a little bit closer, this is times by 100, you can see the images are a little bit bigger. Again, you can really clearly see that structure called the nucleus, those darker circles, that's the nucleus of the cell. And it controls all the cell's activities. It's like the brain of the cell. We're going to go one closer. There is it times by 400. And actually we've even got one closer again, and you can see that structure there of your cell wall and your nucleus. and that cell will be filled with that solution of cytoplasm, and along the inside of the cell wall you have what you call your cell membrane, which controls what goes in and out of the cell. And so this solution here is your cytoplasm. But there's something that is missing from our onion skin cells that is common to most plant cells, and I'm going to show you that in our moss leaf cells now. So here's a moss leaf. You can see it's just been directly shown underneath the microscope in this image here. That's just a picture at the top of a moss leaf. If you can remember in the first video, you can go back to it if you like. I showed you a moss leaf, just a prepared slide of that. So it maybe wasn't quite as bright green because it maybe had been preserved. But we saw some of the cells of moss leaf. But I'm going to show you another picture to show these cells in a little bit more detail. And here they are. So what do you notice that's different? We can see our cell walls really clearly, those kind of divisions between the cell, almost you can see it here. But you can see there are some green structures in these cells, and those are what we call chloroplasts. And again, we'll spend a wee bit more time looking at what chloroplasts do in a later lesson. But chloroplasts are little structures within a plant cell that actually make the plant green. And they contain a pigment called chlorophyll, which traps sunlight during photosynthesis. And that's why plant cells can do photosynthesis, whereas animal cells cannot. We looked a bit at that a bit in our previous topic, our similarities and differences topic. So a moss leaf is different than an onion cell in that it contains these chloroplasts. And have a think about onions. Are they green? No, they're not. And that's because they grow underground. They don't, that part of an onion does not need to trap sunlight. Onions will have leaves above the ground that will trap the sunlight to provide it energy. Whereas a moss leaf cell, it will be above the ground. It needs these chloroplasts in order to be able to trap the sunlight for photosynthesis. So the main difference between moss leaf cells and onion skin cells is that onion skin cells do not have chloroplasts. So if you want to fill that in your notes, just underneath where we drew the diagram of the onion skin cells. So the main difference between moss leaf cells and onion skin cells is that moss leaf cells contain chloroplasts so that they can trap sunlight for photosynthesis, whereas onion skin cells do not have chloroplasts, and that's why we didn't see any green structures within the onion skin cell when we looked at it under the microscope. And that's because that part of the onion will grow underground and cannot trap sunlight, so it doesn't need chloroplasts. So as I said, we're going to look at those structures in greater detail in a later lesson. But that's the end of today's lesson. What I want you to do now is to go back to page 2 and is to spend a bit of time learning your parts of the microscope. So I've written some details on the screen there to help you. So what I need you to go and do now is to learn the parts of the microscope. So focus particularly on your spelling, so the different parts and labelling them in the correct place, but also focus on your spellings. And what you might want to do is to use page 2 and to cover the words that you've written in and see if you can recall them by just saying them out loud. But I have also uploaded a blank copy and the instructions for today's work, which you can find on Microsoft Teams in the Files section. And you can use that to practice. So maybe when you've learnt all the different parts, to practice that, you could print that off a couple of times and try it a few times. And then in your next lesson, so that'll be Thursday for 1S and Friday for 1A, you will do a quick quiz or test on the parts of the microscope in your next lesson, and spelling will be really important for that as well. So that will take the form of a little quick Microsoft Forms quiz, and I'll be able to see how you've got on in that. So spend a little bit of time learning those parts of the microscope so that you could label those really well.